So continuing on with the third one of our protozoans, our third phylum, which are the cercozoans. This is part of a group of organisms referred to as the amoeboid-like ones, meaning that they have these amorphous type shapes, meaning kind of this blob-like or undefined shape behind them. But what's unique about the cercozoans is that they have this little kind of outer enclosure associated with them, meaning like a shell. And that's made out of a compound called calcium carbonate, which is the first out of the two main components of bone. So they have a kind of a bone-like shell. But what's also unique about them is that they also have these kind of perforations as part of their bodies to allow them to eat, to consume, and exchange. And so they get these kind of very neat shapes that are kind of blob-like in these enclosures, but they're full of little foramina or tiny little holes. And hence the usual group of them is referred to as the foraminifera. Now these are really closely related to their cousins, in this case called the Radiolarians, also being referred to as radiozoans, which also happen to be amoeboid-like. They also move through pseudopods, in this case thread-like kind of extensions. These are what we call radially centered or more or less symmetrical. They still have a shell, but in this case it's made out of silica or silicon dioxide, or sand, if you want to think about it that way, which also kind of those unique shapes, except that they're a little bit more symmetrical and part of our zooplankton. So this is the stuff that our fish and our whales and things like that end up consuming in the oceans. So a little bit more symmetrical, but same type of shell-like and odd-like shapes behind them. Now, out of the third group, this is really where we're going to focus most of our attention. The cercozoans and the radiolarians just have this kind of contribution to the biogenic cycle of life. But amoebozoans, on the other hand, the true amoeba, or amoebae, if you will, these ones also move through pseudopods, meaning in this case that they ooze out into large blobs of their body to move. They do not have a shell related to the cercozoans and radiolarians, and they also possess these tubular type mitochondria. Now, the reason why we really focus on these guys is because a lot of them have some sort of pathogenic type origin. And so probably the more important one of them all, which I'll talk about a little bit later, is the two groups that we're interested in is Naegleria fowleri, in this case, which causes a disease called primary amoebic meningoencephalitis, also known as PAM. Or also referred to as amoebic encephalitis, right? And this is commonly referred to as the brain-eating amoeba, in case you've heard that term before. So this guy is the one that kind of makes it to your brain and literally starts kind of poking holes into your brain on its own, right? And so we'll talk about that when we do a little bit of the DXM kind of lecture a little bit later. Now, its other cousin, also part of that, similar kind of situation. In this case, acanthamoeba, which causes a wonderful disease called keratitis. And so here, what we're looking at is the acanthamoebas like to go to your eyes. And so here, for people that like to go kind of swimming in little lakes and open bodies of water in which these organisms happen to be growing, they'll usually go straight to your eyes or through your nose and access your eyes. And it'll start from anything from eye pain to redness and a little bit of blurred vision. And then ultimately, as it starts actually destroying your eyes more than anything else, you become very sensitive to light and eventually start tearing up a lot and then eventually go blind. And so what happens is you get this really strong inflammation into your eyes and you start developing these keratoid type growths onto your eyes. It looks like you're almost having glaucoma. That's really kind of the best look I can describe. And so eventually some of these can actually make it to your brain as well and cause very similar type of symptoms like Naegleria does as well. So those are the two big deals. One causes you to go blind, the other one that causes you to go crazy, if you will. And then the other version of them, which happens to be Entamoeba, one of the ones we're going to look at in our lab, Entamoeba in this case causes the common disease known as Amibiasis. So Entamoeba histolytica is the one that you're kind of really looking at over here. Same idea. It's common to most people that live in the tropics, again, due to just poor sanitary conditions and poor access to clean water more than anything else. Not everybody usually gets sick. Some people can become can be asymptomatic, but in the people that happened to undergo amoebiasis, which is probably the more common disease associated with it, is this kind of heavy, very strong gastrointestinal type of distress. And roughly about 10% of our population, so not a lot. So 1 in 10 people will get really, really sick. And that means they can start getting other side effects from having like ruptures and issues into their liver. It really kind of spreads out and kind of causes havoc in terms of your organs or anything else. So kind of a dangerous guy to play with in there in certain cases. Now, on the side of this, we're going to kind of do a little bit of a left turn here and look at the slime molds, which again, they're currently being reclassified, but under this current condition, these guys happen to be probably one of the more unique protozoans and kind of more fun to study behind them. So I'm going to kind of go through their structures as well as their life cycles, which is kind of important to know, and then you guys can know a little bit more of the fun parts behind them associated with them. So our slime molds here, still amoebozoans, still part of the same phylum, are a group that under the name mold, we used to kind of assume that they kind of were more related to mushrooms more than anything else, hence the term mold itself. But there's no better way to describe them, to be honest. They look like just puke. There's just no other way. If you've ever seen like a little chunk of vomit sitting down somewhere from your cat, from your dog, from yourself, even, that's what these guys really look like. And they get subdivided into two groups, into cellular and acellular versions of them. And they have this very unique life cycles and what they do. Now, the story that kind of associated with this is probably kind of the more entertaining of them all, is because everybody probably has seen a little thing of vomit kind of floating around somewhere until you see it start chasing you. These guys are motile. They move. Remember, the classification of all of these guys is based on the fact that they move. And so slime molds have the ability to crawl in mass. Okay. Now, these guys happen to be fairly innocuous. They don't really harm anybody. They don't really do anything. And as long as there's food in abundance, they kind of are hanging out by themselves. It's only when food becomes kind of lacking or low quantities of them that they all kind of group together and they start moving as a single body. And so this is kind of the two stories that I'm going to give you. So let's look at the first group called the acellular or plasmodial group of slime molds. And here's one of the unique pieces. So normally it exists into this kind of unicellular kind of stage, what we call a haploid stage, so a single stage, kind of like a sperm or an egg. And they're doing their thing by eating their food. But when food becomes scarce, one of the things that happens is that they'll start fusing together, kind of in a fertilizing type of way. But instead of just turning into a diploid organism, which is what we are, right? A fusion of mothers and fathers kind of thing. Here, the zygote starts reproducing and forms this kind of giant mass known as a plasmodium or a cenocytic plasmodium. And really what it is this giant jelly of a bunch of cells all fused together. It's thousands, if not millions, of these guys all into this giant blob. So all the nuclei are all kind of floating around together in this giant blob. Now, what's crazy about that is that that blob moves. And so when it's looking for food, it will start crawling, it will start oozing, it'll start showing, kind of releasing these little tendrils, which is what you're seeing on the top right-hand side, until it finds food. Once it finds food, then it starts assembling itself and it turns into what you guys would recognize almost as if it was like a mushroom, like a little fungus, which you guys know as a sporangium also. And so this is where that term mold kind of came about, simply because they resemble that. And so once they found food, they're happy, they reproduced, so they consume and they start releasing their spores, their children, so they can kind of repeat the cycle. Now, The pause that I'm going to give you here is for you guys to go online and look up slime molds, simply because of the research that is going on behind them, which is very, very unique. We've come to find out that Plasmodial slime molds happen to be relatively intelligent, if you will, even though they're just individual cells. No, there's no brain, there are neurons, nothing like I said before. Remember, in order to kind of have that type of neural development, you actually need something that resembles a neuron, and in bunches. These are individual cells just kind of putting themselves in mass. But what you will see is that they'll coordinate their way to find food, and any generation after that will actually remember. In other words, these cells have memory. And so you can take an organism from this, derive it, and have it go through the same type of searching for food kind of scenario. So what I'm getting at here is that scientists have spent time putting these guys in mazes, kind of like you put a rat kind of thing, and they'll solve the mazes very efficiently. More than anything else, like I said, the generations. Several generations after, the children of the children of the children of the children, and so on and so forth, of these cells can do the exact same mapping. And to the point that this has become such a unique feature of them, that there's videos out there you can look into in which you'll see people testing slime molds as a way to find the most optimum traveling distance between point A and point B. And I mean in terms of the case of you using like your maps on your phone, the same kind of concept. To the point that people have plotted these guys out and they've solved anything from city maps to organization of railways and substations and everything else. These guys can solve them more efficiently than humans can. How? We still don't know. So that's kind of one of the subjects of studies behind these guys. So it's a really, really neat concept. And you can actually see these in real time. Now, their cousins, the cellular slime molds, same concept, same idea. They'll live by themselves, and then they'll move when they're hungry, right? But what's unique about these guys is when they call themselves together, they form a very interesting concept in which when they're hungry, rather than forming themselves into a blob, instead, they turn themselves into a worm. And so here's kind of the more unique part behind them, is that suddenly you'll see this thing that looks like just ooze sitting down on, a piece of dirt, in the middle of a forest, that kind of thing. And it suddenly senses or finds that there's food close by and it's hungry. It'll all kind of move together and it'll turn itself into what looks like a slug. The closest thing I can tell you is a banana slug, for those of you who've been to like Santa Cruz or something like that, is it looks like a giant worm. Well, not quite giant. And so it's nothing kind of short of just awe to seeing something that looks like a blob first assemble itself, turn itself into a fully different structure, and then worm itself up until it finds new food. And then it'll form that kind of fungus-like structure, and then it'll release its spores. That's really where these guys get to be a lot of fun. So videos are plenty available if you're interested to look up how these cellular slime molds kind of aggregate and turn into these cool little worms to travel and find food. So those are really entertaining to find. All right, so we looked at Parabasla, alveolates, our circozoans, Radiolarians, and amoebozoans, all amoeba-like. And then we added the disease ones from the amoebozoans and the slime molds, which are really, really neat. Now, the last two are Euglenids, also under phylum Euglenozoa, which all happen to have this kind of modified flagellum to do with this very interesting turn. And then they're very unique in terms of their mitochondria, simply because the folds inside of them now have kind of disc-like shapes, very similar to what it looks like inside chloroplast, actually. So there's two versions that we're interested in finding. And so here are our Euglenids, which when we talked about paramecium a little while back, and we told you that that was the #1 studied organism on the planet, Euglena happens to be #2. So Euglena used to be misidentified as a plant simply because of its green color and simply because it's photoautotrophic. It happens to have chlorophyll inside of it. So it has the same kind of unique green features that a lot of plants have, simply because of that chlorophyll present. So it's phototrophic. And in the absence of light, it can be chemoautotrophic. So it can produce still its own food and its own energy in the absence of light. Now, what's really cool about this is that these guys seem to have, well, not seem to have, they do have photosensitivity. So they'll go towards the light, very much like plants do in terms of shifting their chloroplasts. And so it does it because it has this cool little thing called an eye spot, which is what you're seeing there that is kind of in colored red. Now, what's really cool about that is that it kind of, in the event that it detects light, it moves away from the light. So it's not like going towards the light, but as it moves away, it goes towards the flagellum and it powers it so it swims towards the light itself. So it has this kind of negative tropism, meaning it goes away from the light so it can power the thing to take it towards the light. So it's a really kind of cool combination behind it. The modified flagellum behind it is something called a paramylon, in which it kind of wraps itself around it, and it has these little granules also that kind of allow it to store food. And so it kind of has this kind of squirmy kind of ability to do that. So it kind of shrivels and extends, and it also powers itself through the regular flagellum that it has. It has these very unique features associated with that guy. Now, the second group of our euglenozoans are the kinetoplastids. So here we take a look at two different diseases, or two simple parasitic type effects that are caused by these. And so the first one that we get to look at here is the trypanosomes, one of the organisms that we do have in under our microscope, also for the lab. And so Trypanosoma, probably the most infamous of them all, is Trypanosoma gambiense, which is something that causes a wonderful little disease called sleeping sickness. And so for those of you who are probably kind of slightly familiar with this, is the sleeping sickness is caused by this really, really cool fly called the tsetse fly. And so more or less the reason why it's called sleeping sickness is that people would get bit by this little fly, the parasite would enter the bloodstream, overwhelm our blood simply because it starts destroying our bloodstream, causing you to get tired really, really fast and pass out. So these are called by two organisms. Gambiense happens to be one of them. Brucei and Krusei all happen to be all species that cause these so-called sleeping sicknesses. And the official term behind that, in case you don't know, is called African trypanosomiasis. Sleeping disease just sounds kind of cooler, right? And like I said, it's insect-borne, so it's caused usually by these cool little flies. And usually what it kind of starts off as this kind of malaise that everybody's familiar with. So you're tired, you have a little bit of fever, headaches, and a little bit of kind of joint pains associated with that. And then at that point in time, as it kind of extends itself, once it truly kind of overwhelms the blood itself, within about 2 to 3 weeks, pretty much you literally faint. And so you don't wake up, you kind of enter this kind of coma-like state, which is where the kind of whole concept of sleeping sickness kind of derives, right? So you usually can kind of see it very quickly because the bites are very, very kind of prominent from this little fly or anything else. But like I said, it gets kind of like a standard disease, kind of get tired, and eventually you kind of fall out and pass out until you can't wake up, hence the term sleeping sickness. Now, the other one that is kind of related to it is caused by a group of organisms called Leishmania or Leishmania, depending on who you talk to, right? And so it causes a wonderful little disease called Leishmaniasis. And also kind of very common in terms of tropical areas, in case you're wondering. And so this guy is also caused by another little bug, several different versions of them, but they're called sandflies in this case. And so it causes two versions of the disease called a cutaneous leishmaniasis, a skin one, and there's also a visceral one in which it kind of affects your internal organs, right? And so the skin one, obviously, as you can tell, is the skin sores that you're seeing over there. The visceral version of this one ends up affecting things like your spleen, your liver, and sometimes in some very, very kind of rare cases, can enter the bone marrow. So this is really where the the big issue is. So for the cutaneous version of them that you're seeing there, so these pretty much are giant sores that kind of start as little papules or little bumps, and then eventually kind of develop into these kind of ulcer-like skin lesions that you're seeing over there. And these are usually painless, although some, in rare occasions, people kind of report pain associated with it, especially if they're kind of associated in places that fold a lot, like in the middle of your skin, like armpits, that kind of thing. But the visceral one is usually the kind of the more dangerous one of them all too, simply because it's kind of relatively silent, it's asymptomatic, if you will. And so once the fever kind of overwhelms the body, and then you start having swelling of the lymph nodes, then we pay attention to the spleen and liver that end up being swollen as well, thanks to Leishmania. And then at that point in time, it usually involves a much serious intervention, medically speaking. So that's where these two guys kind of really are involved in terms of diseases. And then the last one, probably the more infamous of them all, kind of interesting out of our diplos, is the Diplomonadida doesn't really have a lot of the organelles in the sense that we know them. They still have them, obviously, because they can't really exist without them, something that creates energy, something that creates proteins. They just have very interesting versions of them. These are probably kind of early versions of these organelles. They also happen to be binucleated and lots of flagella. And probably the reason why we look at this, and this is also one of the ones we get to see under the microscope, even though probably this is the most difficult of them all to kind of look for under the microscope. It's kind of hard to find most of the time. Most people report a lot of trouble looking for this guy. Thankfully, I'll do that for you guys. Is Giardia lamblia, which happens to cause a wonderful little disease called Giardiasis, which also happens to be another gastrointestinal disease. So lots and lots of diarrhea, if you will, again, caused by usually drinking water from contaminated sources. So this typically is a freshwater type of thing. So ponds, lakes, pools, that kind of thing. And probably one of the reasons behind its infamous name is because of its shape. And so Giardia lamblia happens to have this, again, binucleated look. So when people look at it under the microscope, it usually ends up looking like it has this kind of heart-shaped body with two little eyes. And so you can kind of literally look up Giardia and you'll see tons and tons of people reporting pictures. And what's also really neat is about that flagellum, usually one of them is kind of at the center base of the organism. And so what it looks like is that it looks like it has a little mouth also. And so you end up looking at this organism like it's kind of smiling at you in this little heart-shaped container. So look these up. You'll have some fun with them. If you get bored, you can always send them to somebody on Valentine's Day and wish them Giardiasis or something like that. All right. So that kind of puts together all 7 of our lab-based protozoans that we're interested in looking at, with the last one being our diplos. So this is where we're going to end in terms of this content and enter our next group of what we call our non-microbial parasites, right? And so the idea here is that this is the field of organisms that at some point in time in their stages, they had a microscopic portion. In other words, before they got all large and kind of scary looking, there was at least a reproductive stage, a feeding stage, some sort of stage that managed to get inside of you that was microscopic for a small portion of its time in its life cycle. Okay, so truly here is the entering of the field of parasitology and our larger, what we call ectoparasites more than anything else.